We were just talking about aerobic and anaerobic reactions. Aerobic reactions require oxygen, and they make a great amount of ATP, energy, and they store it in ATP. And this occurs in the mitochondria. Again, you probably remember from biology class that it is the mighty mitochondria that is the center of cellular respiration. Anaerobic reactions do not require oxygen. They are without oxygen, and they make just a little bit of energy and store it in ATP, and this occurs in the cytoplasm. Now we consider glycolysis. It's the first reaction of the glucose breakdown, so just picture a 6-carbon molecule and it's broken in half. It breaks down glucose into 2 pyruvic acids. Those are 2 3-carbon molecules. It occurs in the cytosol, that's the same as cytoplasm. And this is an anaerobic phase of cellular respiration. And remember, as anaerobic, it does produce ATP, but not a lot. So 2 ATP molecules are produced per glucose molecule that you break down. So here is glucose, 6-carbon molecule, and you simply break it down into 2 pyruvic acids right there. In the anaerobic reactions, glycolysis makes much less ATP than aerobic. Glycolysis is anaerobic, and there's a net gain of 2 ATP molecules per glucose that you break down. There is a molecule called acetyl coenzyme A that is produced, and it is the molecule that connects carbohydrate and lipid metabolism. Next is the citric acid cycle, and then electron transport chain is going to occur. And the end product of respiration is production of carbon dioxide, water, and up to 36 ATP molecules for each glucose molecule that's broken down. This is the overview of cellular respiration, and it shows you here that glucose is broken down into 2 pyruvic acids outside of the mitochondria. Here is the mitochondria. So this is showing the anaerobic portion in the cytoplasm, also known as cytosol, and this is showing the aerobic portion. And again, as we said, catabolism, catabolic pathways break down something, a large molecule, and produce energy. Anabolic pathways are for production or for storage of that energy that is formed somewhere else in the body. Glycogen is the storage form of glucose in animal tissue, and fat is used to store adipose tissue. So glycogen and fat are both potential energy stores. We can take the glycogen, break it down, and release glucose for immediate energy. Same thing for fat there. It is storage form of energy, fat. Next is nucleic acids and protein synthesis. So the 2 nucleic acids that we will talk about are deoxyribonucleic acid, also known as DNA, and then ribonucleic acid, known as RNA. DNA is the genetic material. It's found in the nucleus, and it provides instructions for how our entire body will function. Every single aspect of our body is determined basically by DNA. Now, can your environment definitely affect that? Yes, it can. DNA, RNA, and protein molecules carry information because they consist of sequences of building blocks, and it's constant recombination of those sequences that make us who we are. The genetic information, that's instructions that tells the cells how to make proteins. The gene is a section of the DNA that has the information for making one protein. The genome is a complete set of genetic information in the cell. The exome is a portion of the genome that codes for a protein. This has also been called an exon. And then gene expression is our ability to make certain proteins in each individual cell that makes us who we are. A nucleotide is a building block of DNA. It is made up of a sugar, a phosphate, and a base. In DNA, that sugar is deoxyribose. then it's connected to a phosphate, and then directly connected to the sugar is the nitrogenous base. Our bases are A, T, G, C. In DNA, that is adenine, thymine, cytosine, and guanine. The shape of DNA is a right-handed double helix, that is, a ladder that's twisted to the right. And the bases that bond to each other are always A to T and G to C. Adenine bonds to thymine and cytosine bonds to guanine. A to T, G to C. And here you see the sugar backbone, and then on the outside here are phosphates and the base attached to each sugar, and they are bound together, A to T, G to C, and we have alternating, we have hydrogen bonds that connect those bases together. So again, it's a double helix. The sugar in DNA is called deoxyribose. The bases in DNA, A to T, G to C, and they're called complementary base pairing. DNA wraps around histones in order to form chromosomes. And this shows you the DNA, right-handed double helix. It folds and folds and folds, and it then produces a chromatin molecule. The chromatin continues to fold and produces chromosomes. Nucleic acid components going from small to large. Here are the nitrogenous bases. The nitrogenous bases are a part of nucleotides. Nucleotides are made up again of sugar, phosphate, and a base. Then sections, large sections of the DNA comes together to make a gene that codes for particular proteins. And then those genes are all found on chromosomes, and all of the chromosomes, all of the genetic material comes together to make up our entire genome. DNA profiling frees a prisoner. That is we have been using DNA profiling in order to determine guilt or innocence in criminal cases. The human genome contains 3.2 billion, with a B, bits of information. Much of that genome does not actually encode proteins. Much of it is referred to as junk DNA, which don't really like that term because there's a lot of things we haven't actually discovered the purpose for yet. So DNA fingerprinting actually compares the most variable parts of the genome among individuals in order to identify individuals. So we use those for crime scenes, for paternity, and other such criminal cases. Okay, next, PCR, polymerase chain reaction, is actually a lab version of DNA replication. It speeds up the process of DNA replication in order to produce amounts of DNA that can actually be tested in the lab. Now, what is DNA replication and when does it occur? Every time the daughter cell divides, it must have identical copies of DNA, and that process is called DNA replication. It occurs during interphase, and the first thing that's going to happen is the hydrogen bonds are going to be broken in the middle. The strands are going to unwind and separate, and each side is going to get a corresponding new side, and in the end, you're going to form two new helices. So two double helix molecules are formed. Helices is the plural of helix. Now, the genetic code. The genetic information stores the correct sequence of amino acids for a polypeptide chain. So it's a correspondence between the DNA nucleotide and the amino acid that it is going to code for. Each of the three bases in DNA is going to code for three DNA bases in the RNA, and each of those three is called a codon. That codon then codes for particular amino acids. Those amino acids are going to be put together to make a protein, and we have our hair color, eye color, skin color due to the proteins that are made in our body that are determined by our DNA sequences. Okay, so codons are each of the three nucleotides that codes for an amino acid. RNA molecules are single-strand nucleotides, and there are some differences in DNA and RNA, and we'll go through those in 1 second. Transcription is the process of copying the DNA and making an RNA sequence. You have messenger RNA that's found in the nucleus. It makes the copy of the DNA and that goes out throughout the cell in order to carry the message of the DNA. Now, the differences, basic differences in DNA and RNA. DNA is a double strand, RNA is a single strand. DNA is found only in the nucleus. RNA has the ability to travel outside the nucleus. Nitrogenous bases of DNA include ATGC. Nitrogenous bases of RNA include adenine uracil. That's a different base than cytosine and guanine. So uracil replaces thymine in RNA. Now, again, this is an overview of protein synthesis. We're only going to get the overview in anatomy and physiology. I know that you have gotten this in biology, but here's the basics. DNA is that right-handed double helix. It stays inside the nucleus. It untwists, it unwinds, and messenger RNA is made as a series of complementary bases to that one strand of DNA. So for every T, you're going to have an A, a G, you're going to have a C. So again, it's going to be ATGC, except in this RNA strand here, there is no thymine, there's uracil. So you look right here. The DNA has adenine, well, the corresponding base in RNA is uracil. Okay, so you're going to make a copy, and that is called messenger RNA. It's going to leave the nucleus, go out into the cytoplasm, and each of the three bases on messenger RNA is called a codon, and it's going to get one of these transfer RNA molecules to bring in an amino acid. The transfer RNA molecule is going to bond each of the three bases on mRNA. It's called a codon. The three bases on tRNA is the anticodon, and it allows that tRNA to line up just as it is mandated by the DNA in our bodies, in our nuclei of the cells, and it's going to come in and drop off one amino acid and another and another and another, and those amino acids are going to come together to make a protein. And protein synthesis is called translation, and that occurs here on the ribosome. So codons, again, are made up of three nucleotides. Anticodons made up of three nucleotides. But codons are part of the mRNA. Anticodons are part of the tRNA. Protein synthesis. There's 20 different types of amino acid. There's a total of 64 different combinations of these codons. But we only have 61 codons that actually specify the 20 amino acids, because 3 codons are actually stop codons. You have a start codon, also called an initiation codon, and it codes for methionine, and it signals the start of a protein. So AUG codes for methionine, and it's also called a start codon. So there's a total of 64, but only 61 of those actually code for the 20 amino acids. A stop codon is encountered whenever that is encountered, protein synthesis stops. A peptide bond. Peptide bonds are formed between adjacent amino acids, and they hold those amino acids together. A chaperone protein. It helps a protein to fold. It guides a protein in the folding process. Now, the different codons here, this shows you're going to have several quiz questions on this, and on these quiz questions you see here U, and then look on this other side U codes for phenylalanine. So you read each of these bases. This is the first letter, across the top is the second letter, and down this side's the third letter. So UUA is going to code for leucine. So you can use this chart in order to help you determine the bases that code for different amino acids. Transcription is copying of DNA into RNA. It occurs in the nucleus. Translation is the production of a protein, and it occurs on the ribosome. 99.9% of the human genome are the same among all people. We only vary in 0.1%. 3 genetic changes are mutations, SNPs, and changes in copy number. And then what is a mutation? It's a change in the DNA sequence. Some are spontaneous and